So, tunica interna, tunica media, the middle portion of the vessels. It contains smooth muscle. What does that bring to mind? You're thinking about viscera, and do you happen to does anything come to mind about, do we control its activities? No. Okay? Smooth muscle is under involuntary control. Therefore, it's innervated by the autonomic nervous system. Wait a minute. My blood vessels are innervated? Most definitely. Because they have to have the ability to either constrict or dilate. And the smooth muscle is going to allow that to happen. We have some collagen because that's some of the fibers that helps to build them. And depending on where and what vessel we're looking at, elastic tissue could be present. One of the things about their structure, you're going to find that when we look at these vessels, some will be thicker, some will be thinner, it's really going to depend upon what you're looking at in what area of the body, and are we taking blood away from the heart or bringing it back, okay? Many different things are going to affect what tissues are present. In looking at the structure of the medium, the middle portion, it does help to strengthen the vessels, especially arteries, and it prevents blood pressure from rupturing them. Ever seen a video or anything of an aorta, right there at the heart, rupturing? YouTube it sometime. You will be shocked. Ever seen somebody like giving blood and their pressure is so great that it spews the needle out and the blood flows across the room, flies across the room? Pretty cool, but you know, it's anyway. The other part, because like I just said, we have smooth muscle, it's going to regulate the diameter, meaning constriction or relaxation, the dilation. The external portion, loose connective tissue. This allows for merging with either a neighboring vessel, a nerve, or organs. It also helps to anchor the vessels in the body, helps provide passage for the nerve that needs to come in and innervate the middle portion. I will also find that I'm going to be dealing with my lymphatic system because that's going to be vascular also. And then we're going to have vessels that have to supply the vessels themselves. That's called vasa vasorum. Yeah, did everybody understand that? I got to have vessels to feed the vessel itself because it needs a blood supply. It's tissue. And in the vessels, the smaller ones that feed the larger ones are termed vasa vasorum. It's a really cool structure. The arteries, these are also known as resistance vessels. They are very muscular and they can withstand the surge of blood. Every time the heart beats, for arteries, they are under pressure. So, we're going to say that the size of the artery determines what class they're in. A lot of times when we think about the blood vessels and how materials are going to move, this is pretty much what we tend to think about. We tend to think about this large structure, these larger structures, I should say, that we tend to see, all right, like under the skin and that sort of thing. The way that it's going to work, these large ones that we do think about, okay, the ones that have the names, okay, like the brachial, the radial, the femoral, all that sort of stuff, okay? But those are called conducting. That simply means they are carrying the blood somewhere. All right? They're large. They're usually very elastic in their makeup, and they simply are directing the blood flow somewhere. When we begin to get to having blood go to a specific organ, i.e., blood moving to the kidneys, okay? It has now turned into a distributing artery. So it came off of this large one, the descending abdominal aorta, and it branched to one kidney, branched to the other kidney, and that one was my distributing, because it took it to a specific area. Once we get to the tissue itself and we begin to have all of the little branching that's going to occur, I've now reached resistance vessels, the small ones. These, which are usually microscopic, I'm just trying to point to an area of the tissue. Those are my arterioles. They are the ones that are in control. That means the very smallest of my arteries, the part that's actually microscopic, that I can't see, is in control. Let me repeat that. My arteries that are going to start out huge, exiting the heart, the aorta. As they are large, elastic, can withstand the blood pressure, they can expand and recoil, expand and recoil. They'll have the ability to direct my blood flow. Branching will begin to come off. We'll see that we're going to have branching go to a specific area, i.e. the kidneys, by way of a renal artery. Okay? Once it reaches into the tissue of the kidney, and I enter arterioles, I am now going to be at the level of control. The only thing the larger arteries did was direct my blood flow. It is going to be what happens at the smallest level where I get the control. Now, when I say control, meaning, is there going to be blood flow to that tissue it meets? Is that tissue that the smallest artery meets going to receive any blood flow? In some cases, we're going to get even smaller. We can have, in some cases, met arterioles. They can actually help link that arteriole, the smallest portion, to the capillary bed. They don't have to be present everywhere. It's just certain things. This will allow, in the case of the met arteriole, we get a shortcut and blood gets to bypass a capillary altogether. That means if the blood bypasses the capillary altogether, there was no blood flow to that tissue at that capillary bed. It went straight from an artery to a vein. Does it make sense? Okay. The way that we're going to monitor blood pressure, I mentioned these a ton of times. Not going to spend a whole lot more time on them. In the arteries, I have sense, meaning sensory organs. The ability to pick up information. So they are termed arterial sense organs. Their job, monitor blood pressure and my blood composition. What do I mean by blood composition? A little bit. What?
I said like what it's made up of.
And what kind of stuff?
Like the small elements, the protein, like the red blood cells, the platelets.
Those would be able to affect what that blood composition would be monitored for, because that blood composition is monitoring what that means. It's monitoring gases, stuff like little bits of waste, some of those electrolytes, okay? pH, all right? So, why do I need to be monitoring that information?
We don't get too much of it.
So that I am making sure, for example, pH is not too high or too low. Because if that's the case, I'm affecting the function of all the cells of my body. So blood composition, it's looking mostly at the gases and the pH.